In this review, we are going to be taking a look at the structure of DNA and also looking at the basic replication mechanism involved in this for this particular macromolecule. So let's go on ahead to our first slide, just looking at the basic structure of DNA and another macromolecule of the same type, another nitrogenously based molecule, the nucleotides, and or the nucleic acids more broadly. The DNA and RNA, they have a similar structure. They are made up of a sugar phosphate backbone, and then they have these nitrogenous bases, these A's, C's, T's, and G's that you see here that have a lot of nitrogen within them. One of them is shown in more detail here on the right, and the thing or the part that's different about them is this nitrogenous base, and again you can see lots of nitrogen within each of those. If you take a closer look, this is actually thymine, so here's that sugar and here's that phosphate. That's the same for all of the nucleotides, the A's, C's, and the T's and the G's. But the part that differs again is this part that sticks out sort of to the side in both DNA and RNA. DNA is a little different than RNA. DNA is double-stranded, so you'll see these A's pairing up with T's and C's pairing up with G's in the center of the molecule. And just to give us a closer look at these nitrogenous bases, we have our T, or thymine, and our C, our cytosine. And then we have A for adenine and G for guanine. And again, when we're talking about DNA, we're talking about A pairing up with T. So we always have one of these purines pairing up with pyrimidine. And we have a guanine, G, pairing up with a C, a cytosine. Okay. Now, in RNA is a little different. Instead of having a thymine, we have a uracil. That is a big difference with RNA. Also, RNA is single-stranded, which we're going to talk a little bit more about RNA as we move through the chapters. So, just to give us a quick overview of this structure, here is DNA, double-stranded. So you can see the sugar phosphate backbone here in the center with the nitrogenous bases pairing up in the middle. And they pair with very weak kind of bonding called hydrogen bonding. And that bonding in the middle there is actually something that facilitates or is a function of the replication process. So if we take a look at the actual replication process, we're showing here just a real basic look at the DNA. It's not twisted as it usually is into a helix. For simplicity, it's just shown here kind of as a ladder, and we see our nitrogenous bases pairing up. And again, because there's very weak bonds between these nitrogenous bases, they're very easily separated. So you can see those two parent strands in blue. And then during the replication process, the new nucleotides are brought in by an enzyme. So if the enzyme sees an A, it'll bring in a T. If it sees a G, it'll bring in a C, and so on and so forth, until we have two identical molecules at the very end of the process. So that really is just a general overview of the replication process and the structure of DNA itself. In the next few videos, we will be talking about how the information in DNA is actually used to make proteins. So this is called expressing your DNA or expressing your genes. So we have chromosomes, which have many genes or units of DNA that code for particular proteins, and the overall process flowing from the DNA, the process of copying the DNA into a different kind of a language, that RNA language, single strand. So making a copy of one of the strands or the other, depending on the gene. and then that RNA leaves the nucleus, goes into the cytoplasm, if we're talking about a eukaryotic cell, of course, to a ribosome where the process of translation occurs. And that's where we take RNA and we put together amino acids to make a protein, so expressing the DNA. All right, so I hope this has helped you understand that basic idea or basic topic of DNA structure and replication. Please let me know if you have any questions.